Hello, my name is Lauren Frederick, and I will be presenting the research article Skeletal Muscle, Review of Molecular Structure and Function in Health and Disease, written by Kathvia Mukund and Shankar Subramaniam. We'll explore the fascinating world of skeletal muscle, one of the most essential and adaptable tissues in the human body. Skeletal muscle is responsible for voluntary movement, posture, and even plays a significant role in metabolism. Muscle health is closely tied to systemic diseases, aging, and even neurological conditions. Recent advances in molecular biology and omics research have allowed us to understand skeletal muscle on a deeper level than ever before. Let's explore this tissue from development to disease. Let's start at the very beginning. Muscle formation, or myogenesis. This is a highly regulated process where muscle precursor cells, called myoblasts, mature into fully developed muscle fibers. Specific genes and proteins act like instructors to guide the growth and repair of muscle tissue. Key players include MyoD, MyF5, myogenin, and MRF4, proteins that tell myoblasts how to differentiate and form muscle fibers. Those are all listed right here on this infographic, and I'll talk more about that in just a moment. There's something even more fascinating. Muscle has its own built-in repair system. Satellite cells, which is a type of muscle stem cell, activate when damage occurs, like after exercise or injury. These cells multiply, fuse with muscle fibers, and help in regeneration. However, as we age, this repair process slows down, which is why injuries take longer to heal in older adults. So maintaining muscle health isn't just about strength, it's about ensuring proper repair and regeneration. This figure represents the major transcription factors involved in muscle development and shows the temporal sequence of activation across various stages of myogenesis. Satellite stem cells expressing PAC7 derive from PAC3, PAC7 expressing progenitors, like this precursor cell. Whereas satellite myogenic cells additionally exhibit an activation of MyF5. Following the activation and entrance into the cell cycle, stem cells express MyF5 and MyoD1. Activation of MyoG and MEF2C with downregulation of MyF5 and later MyoD1 marks the start of terminal differentiation. Activation of MRF4 happens several days after the induction of differentiation following a reduction of MyoG. Now, how does the nervous system control our muscles? This happens at the neuromuscular junction, a critical communication bridge between motor neurons and muscle fibers. When a nerve impulse reaches the neuromuscular junction, it triggers the release of a chemical called acetylcholine. This chemical binds to receptors on the muscle, creating an electrical signal that tells the muscle to contract. But what happens if this system fails? Conditions like myasthenia gravis, where the immune system attacks these receptors, can lead to a severe muscle weakness. This highlights the importance of neuromuscular junction integrity in everyday movement and even in disorders like ALS, where nerve-muscle communication is progressively lost. This picture is a representation of the neuromuscular junction and its main molecular actors. The three specific regions define the neuromuscular junction: the presynaptic, synaptic space, and postsynaptic. The presynaptic motor nerve terminal, where vesicles fuse with the terminal membrane to release acetylcholine. That's here with green dots. It's going to release them into the synaptic cleft here. Calcium influx through the voltage-gated calcium channels, so here, trigger vesicle fusion and release from the active zones. And the synaptic space in the middle, my laser pointer didn't work. So here's the acetylcholine in green and the calcium channels here. Here in the middle we have the synaptic space. It contains the basal lamina, that's kind of this gray line here in the middle. And it shows the presence of acetylcholine E and CoQ, which is essential for the inactivation of acetylcholine. CoQ over here binds to MuSK here, and perlecan necessary for the stabilization of the basal lamina. MuSK enables ACH, acetylcholine R, clustering via rhapsin here. The postsynaptic organization of the skeletal muscle membrane includes several folds with receptors for the diffusing acetylcholine at the crest and voltage-gated sodium channels at the trough. These are for efficient neuromuscular transmission. All right. Now let's zoom into the muscle fiber itself. The sarcomere is the fundamental unit of muscle contraction. It's like a microscopic machine made up of actin and myosin filaments. These filaments slide past each other to create movement powered by ATP, which is the energy currency of a cell. Proteins like titin and nebulin help maintain the structure and elasticity of the sarcomere. Think of them as the scaffolding that holds everything in place while movement happens. When mutations occur in these proteins, we see diseases like muscular dystrophy, where the muscles progressively weaken due to improper contraction mechanics. My sister actually suffers from this, so I see this all the time with her, and we work very hard to try to maintain her current muscle strength. So in our top diagram, this is a representation of the striated skeletal muscle sarcomere, showing the arrangement of thick and thin filaments in the sarcomere and identifying the bands of overlap between them. In our bottom picture, we have the sarcomere summarizing organization and location of major sarcomeric proteins. Systolic calcium brings about a conformational change in the structure of troponin C down here, revealing myosin binding sites. So myosin here will bind to the troponin C. Myosin heads successfully bind and crawl along the length of actin. So along this line here, these will bind together and then crawl as they're contracting and relaxing. The whole host of proteins within the M line here and Z disc function mainly to maintain structural integrity of thick and thin filament lattices. The Desmond intermediate filaments here reinforce and integrate the structure of the muscle cell by forming transverse links between adjacent myofibrils. Muscle isn't just about fibers. It also has an important support structure known as the extracellular matrix, or ECM. This network of collagen, laminin, and fibronectin provides stability, aids in force transmission, and helps regulate muscle repair. When the ECM becomes overactive due to chronic injury or disease, fibrosis occurs. That is where excess connective tissue replaces healthy muscle. This stiffens the muscle, reducing its ability to contract properly. So while we often focus on muscle fibers, the ECM is just as important in maintaining healthy movement. This picture is a representation of the main extracellular matrix proteins and their approximate localization surrounding skeletal muscle. We've discussed before the basal lamina here, the space between the neuromuscular junction and the muscle. It is responsible for inactivation of acetylcholine and holds key proteins that is necessary for stabilization and binding of neurotransmitters. In the extracellular matrix, collagen provides structural support and tensile strength. Laminin, which is here, in orange, it acts as the glue connecting cells to the matrix and facilitating cell signaling and migration. And then fibronectin, which is these green bundles. It also serves as a scaffold organizer, promoting cell adhesion and migration and influencing tissue repair. Do you ever wonder why endurance athletes have such high stamina? One key reason is their muscle's vascular system. Skeletal muscle is densely packed with capillaries that deliver oxygen and nutrients needed for sustained movement. The key player here is VEGF. It is a growth factor that promotes new blood vessel formation. During exercise, your muscles send signals to increase VEGF production, leading to better oxygen delivery over time. However, in conditions like diabetes or peripheral artery disease, blood flow to the muscles is impaired, leading to fatigue and poor healing. This highlights the critical role of proper circulation in muscle performance and recovery. Here we're going to talk about muscle stress. Inflammation and oxidative stress are major contributors to muscle fatigue, injury, and disease. Inflammation is a normal response to exercise and injury, but when it becomes chronic due to conditions like obesity, aging, or autoimmune disorders, it leads to muscle degradation. Cytokines like TNF-alpha and IL-6 drive this chronic inflammation, breaking down muscle proteins. Similarly, oxidative stress occurs when harmful molecules called reactive oxygen species, or ROS, accumulate in muscle cells. These molecules damage proteins and DNA, leading to muscle dysfunction. Fortunately, antioxidants, either from diet or the body's own defense mechanisms, help counteract this damage. This is why maintaining a healthy lifestyle with proper nutrition and exercise is crucial for muscle health. This picture is a representation of the relationship between inflammation and oxidative stress. There are various triggers, including excessive exercise or a complete lack of, nutritional excess and deficits, or disease and injury, which greatly influence the mediators. Muscle pathophysiology. Let's look at the 3 key changes that muscles undergo in response to different conditions. First is atrophy, or muscle loss. It occurs due to inactivity, aging, or disease. The body activates the ubiquitin proteasome system and autophagy, which breaks down muscle proteins. Next, we have hypertrophy, or muscle growth. It's driven by resistance training and signals from IGF-1 and the mTOR pathway, which boosts protein synthesis. Last, we have fibrosis, which we touched on earlier. It happens when chronic damage leads to excessive extracellular matrix buildup, making muscles stiff and less functional. Understanding these processes helps us develop therapies for muscle-wasting diseases, age-related decline, and even recovery strategies for injuries. This here is an image of components of muscle structure and function. It's a representation of various functional components necessary for or arising as a consequence of muscle function in health and disease. The structure and function of each of these units are discussed in the current review. So the arrows identify a one-word description, so injury, support, energy, oxygenation, protect, again injury here, regeneration. Those are all showing the role in governing normal muscle function. So what's next for skeletal muscle research? With advances in genomics, bioinformatics, and systems biology, scientists are uncovering new molecular targets for muscle regeneration and disease treatment. From gene therapies to regenerative medicine, the future of muscle health looks promising. Skeletal muscle isn't just about movement. It's a complex, adaptive tissue that plays a vital role in overall health. Whether through exercise, nutrition, or medical interventions, maintaining muscle health should be a lifelong priority. Here is the reference of the article that I reviewed for this presentation. Thank you.